My name is Jonathan Nasi. I'm the Director of Translational Science at Inscopix. We're excited to have you join us from your virtual offices at home, as it's especially important to stay scientifically connected during this period of social distancing. You can send us questions via the chat window throughout the presentation for up to 5 minutes after the presentation is over. And we'll do our best to get to most or all of the questions with a hard stop right at 12 p.m. Pacific time. Today, our research scientist, Kevin Zitelli, will be presenting a Nature Neuroscience publication from Susumu Tonegawa's lab at the Picower Institute of Learning and Memory at MIT. Kevin got his master's degree at the University of Pittsburgh in Aaron Giddis's lab, where he focused on the role of globus pallidus neurons in the motor deficits of a Parkinsonian mouse model. Together with his 3 years of research experience at Inscopix, Kevin has strong expertise in imaging, electrophysiology, and behavioral techniques. And so with that, I will hand it over to Kevin. Thanks.
All right, thanks for the introduction, Jojo, and thank you all for joining us. We'll be discussing this paper, as Jojo mentioned. Hippocampal neurons represent events as transferrable units of experience. Key word in that title being events. So just bit of introduction here, just thinking about kind of the phenomenon of daily experience in terms of how that's kind of represented in the brain. The authors here just give us this introduction, an example of kind of thinking about going out to dinner. You go to a particular restaurant, and there's various courses, and so there's different sensations as far as tastes and smells and sights depending on where you are, and so all of these things we kind of know are represented in the brain, in the hippocampus. So things like a spatial map in the hippocampus, certain cells kind of map to a particular spatial location, and they increase their event rate in response to a particular spatial location. And this is called rate remapping. It's been known about for a while. So there's these kind of continuous variations that kind of give you information about your immediate experience that are changing continually. But there's also kind of a higher level of abstraction from that. So as you proceed, from the appetizer to the main course through dessert and coffee, it's all one continuous event, but each one can kind of be thought about episodically. So that if you go out to dinner again another night, and you proceed through the same 4 courses, but it's a different restaurant. You're in a totally different place. You're having totally different food. You might be with different people. But as you progress through these 4 courses, you kind of know what to expect. The appetizer might consist of different food, but you still recognize it as being an appetizer, and that helps you understand what's going to come next. So you can still kind of understand these abstract sequences of events by their kind of abstract commonalities, even if the continuous variations of place, time, stimuli are different. You can kind of phenomenologically just understand the abstractions and how they relate to each other. So as I mentioned, you can kind of break up these continuous variables into discrete episodes. And there's been some evidence from like human blood flow imaging studies that this kind of abstract episodic kind of experience is represented in the brain. And in mice, it's been known for a while that various continuous variables are represented in the hippocampus. But what about this kind of more abstract level of experience? So just briefly, bullet points from the abstract of this paper. So they're looking at calcium activity in dorsal CA1 of the hippocampus. They had the mice do simple tasks where they just ran laps around a simple maze, and every given period, they were given a reward. And they found that, in fact, a subset of neurons that they monitored were modulated by lap number. So they were consistently activated on a particular lap with respect to when the reward was given. And they termed this event-specific rate remapping. So it's analogous to rate remapping that is related to spatial location, for instance, but this is tracking the abstract sequence of events. And they found that this actually persists across various main shapes and lengths, and is separately manipulable from spatial activity. So it seems to be not related just to this any continuous variable that they were able to vary, but it is actually, in fact, this abstract relationship between the different laps in the trials. So let's get into some more detail. So as I mentioned, doing the surgical prep to enable calcium imaging in hippocampus, used Invista microscope, of course, injecting GCamp in dorsal CA1, and implanting a microendoscope above CA1 to monitor calcium events, looking at change in fluorescence. And this is the kind of standard task design, as I briefly touched on when we were discussing the abstract. So it's just a simple square maze. The mice are given a reward in this box at the start of lap 1. And they must run 4 laps around the maze. And then the trial will repeat. So one trial consists of 4 laps. The trial will repeat, and they will receive another reward at the start of lap 1 again. Looking at the calcium activity, they saw what you would expect based on what we know about the hippocampus, and that is that this rate remapping related to spatial location occurs. So just to orient you to this sort of figure, because this will be repeated throughout many of the figures. So these are some examples of neurons that exhibit spatial remapping. So the bottom of this graph corresponds to a particular spatial location on the graph. And you can see that these neurons throughout the various trials become preferentially active in a particular location in the maze. These are just 2 examples here. Some cells were activated purely by reward. So you can see here they're activated in the box where the reward is given and only on the first lap. So that is exactly when the reward occurs. These cells were excluded from any further analysis. And hippocampal cells are also sensitive to, of course, speed, like walking speed, and head orientation. So this is just examples of 2 cells and showing that their activity correlates to walking speed and head orientation. And they use this later on to be able to generate a linear model for cells to be able to correct for differences across trials, between speed and orientation, so they can try to isolate the experimental variables that are changed. But so as I mentioned, a subset of cells appeared to be modulated by lap number. So we see here delta F over F heat maps for these cells that display this event-specific rate remapping phenomenon. So ESR is the abbreviation used there. So we can see that, this is the same as the graph we looked at earlier, where the x-axis kind of corresponds to the location within the maze. And so we can see these cells do also demonstrate this spatial rate remapping phenomenon, but also, these neurons, for instance, although they have this a spatial rate remapping, they clearly seem to only activate on lap 1. Or they are very much preferentially activate on lap 1. Same thing. So these neurons, spatially rate remapped, but clearly prefer lap 2, and so on. So that's kind of the basics of the phenomenon we're looking at, and we'll be examining this in lots of different kind of experimental conditions to try to better understand this. So these are examples of individual neurons. So each individual example of a lap 1, a lap 2, lap 3, lap 4 cell. So we can see here, for example, this lap 1 cell is pretty specific for lap 1. It doesn't activate in the same location, especially like 100% correspondence on every single trial. We can see that sometimes it does in fact activate on lap 2 or lap 3 or lap 4, but by and large, it's almost entirely lap 1 that it clearly prefers. Kind of similar example here with lap 4, where you can see that lap 4 is kind of preferred the most, but it does activate somewhat on lap 3, for instance. So there's some stochasticity, but it is by and large reliably preferential based on lap number. So this is just the exact same figure we were just looking at. I just put it here for reference. So this is where I'm just going to explain briefly the model correction. So as I mentioned a couple slides ago, the hippocampal neurons are also sensitive to walking speed and head orientation. So they generated a linear model to be able to correct variations in those variables to kind of subtract those out. So you can kind of generate how much of the delta F over F is explained by this model that accounts for walking speed and head orientation, and just be left with the unexplained, delta F over F related to motion correction. So that's just a way to kind of account for variations trial by trial in those experiments. So we can see here mean change in delta F over F, whether before and after model correction for one lap 3 cell. So we can see here the lap specificity at the peak. And this cell is clearly preferentially activated on lap number 3. So this is just looking at the proportion of lap 1, lap 2, lap 3, lap 4 cells. Lap 1 is clearly pretty well enriched compared to the other 4. But so this accounts for about 30% of cells that they monitored in the hippocampus. So just to kind of test and see that this isn't some kind of artifact, that it isn't somehow some kind of continuous variable, that it is actually a phenomenon that's related to these episodic experiences, they essentially made all the laps indistinguishable. So there's still 4 laps per trial here, but a reward is given after every single lap. So distinguishing between 1, 2, 3, and 4, that relationship is essentially abolished because the animal doesn't have to count. Well, not that the animal is necessarily counting exactly, but the animal doesn't have to run 4 laps for every reward. They just have to run around the maze once, and every single time they get a reward. So the kind of the salience of lap number disappears. And in fact, we also see a diminishment of this event-specific rate remapping phenomenon. So now less than 10% of cells have a kind of a lap preference at this point compared to when the reward was only given at the start of the first lap. That's kind of the basic outline of the phenomenon. There are these cells that demonstrate this event-specific rate remapping. And what kind of going forward from here is kind of demonstrating further details about this phenomenon and other controls, just to make sure that it isn't an artifact from a continuous variable. So here we're looking across days. So this is an example where they just did the same trial just 2 days in a row. And they're looking at the same cells, individual cells across days, and looking to see how well conserved this event-specific rate remapping phenomenon is. So this is an example of a Lap-4 cell. As you can see here, clearly more preferentially activated on Lap-4 on both days. Also active in the same location in the maze. And here we have the model corrected mean delta F over F on each lap. And this orange number is the Pearson's correlation. So on both days, very well correlated. This phenomenon is conserved. This is just a summary figure looking at all the cells that were measured. So here, this is similar to what we saw before, where the track is represented on the bottom and the individual cells. So the spatial activity is conserved, but also this event-specific rate remapping is conserved as well across days. How do we know that this isn't just a continuous variable like distance traveled? So the animal has to run around the lap 4 times, but how does it know that it's not a lap? How is it not just, maybe it's just ticking off, total distance traveled until the next reward, right? So is this really a continuous variable, or is this actually a discrete event? So in this case, the mice went through a kind of pseudo-random maze elongation experiment. This first case where it's 4 short laps is identical to the first couple figures we looked at. But then this trial might be followed by a trial where the first 2 laps are longer and the next 2 are short. And that might be followed by the first 2 being short and the second 2 being long, or all 4 of the laps being lengthened. So this is just continuously shuffled for, I'm not sure how many trials, 15 or 20 or something like that. So the idea here is that there's still 4 laps, but the laps aren't all equal in distance anymore. So is this event-specific rate mapping conserved, or will we see some kind of bleed through? So this is an example of one Lap-2 cell in each case. So we can see here it maintains its spatial activity, and here's the ESR activity in each case. So this is kind of the standard case, the 4 short laps. You can see it does prefer Lap-2. We can see here now Lap-2 is lengthened, and so is Lap-1. but it still does prefer lap 2. Here, lap 2 is short again, but 3 and 4 are longer. Again, so this is still conserved. And here, again, all 4 are long. We see still lap 2 is preferred. Just a summary of this. So this is comparing the various maze configurations. So the standard versus the first 2 being lengthened, standard versus the second 2 laps being lengthened, standard versus all 4 of them being lengthened, and so on, and all the various comparisons. And we can see that, so this is correlating basically, lap 1 cells in this case versus this case. We can see that actually the cells do conserve their ESR activity preference for a given lap. So that suggests that it is in fact tracking events, meaning basically laps, as the fundamental units of experience here. But to further convince ourselves, we'll vary the maze in a couple of different ways, actually. But kind of the next way is, again, we're looking across days here. So we have our standard maze setup again, 4, square laps. Second day will be still 4 laps, same event structure, but this time the maze is circular instead of square. So the spatial relationship is totally changed. We can still recognize the same lap number and the kind of the lap structure. Even the spatial trajectory is the same as the mouse is running counterclockwise around the maze. So what happens here? So an example of a lap 1 cell here at the bottom, where we have a square trial and a circular trial. So we can see here that it seems like most of the activity is still concentrated in lap 1 across both days, even though the maze is totally different. And in fact, the spatial activity in the maze is in totally non-analogous locations. So it seems like the spatial activity has been totally remapped. But this 1 cell is maintaining its preference for lap 1. Kind of looking at the summary here. So we're looking at the first day, the spatial rate remapping for all the cells. And then tracking those same cells on the second day, we can see the spatial remapping is totally abolished. So that doesn't really correlate anymore, but the ESR activity still correlates for each of these. So we can see here also the correlation across days is well conserved for each of these. And the spatial correlation is not at all convincing. So that suggests that it's able to this ESR activity is kind of able to survive this this change in the spatial location, but the mouse learned this ESR, this event relationship, and that when that was conserved, that was able to transfer between days. This is another way to kind of test a similar thing. So instead of changing the maze from a square to a circle, they're changing the direction the mouse runs across around the maze for certain laps. So basically, this is still the standard case where the mouse is just running counterclockwise the entire time. On the second day, they would run through this shared center part of the maze, but then sometimes they would have to turn left and other times they would have to turn right, but it's still identifiably a lap, right? And it's actually still the same distance traveled. So do neurons maintain their lap preference whenever they're turning left instead of turning right is kind of the question. And it seems like they do. The cells have to kind of change their spatial preference so whenever they have to turn left or right. But in fact, they maintain their preference for lap number, even though the laps are changing in their spatial trajectory. Again, so these were just individual examples. You can see lap 1 here is still preferred, so on for each of the various laps. Here's summary data, again, looking at the correlation between day 1 and day 2. And this is the proportion of cells. So that had a spatial preference that was not on the shared part of the maze, meaning that it was either in like one of these corners or the other. How many of those neurons shared their ESR preference across days, and you can see that a large proportion were significantly preserved. So this is similar to what we saw in the very first figure, where we're going to be abolishing the spatial relationship or the sequential relationship between these laps. But in the first case, we looked at mice that were only exposed either to this case or this case, where the reward was given once every 4 laps or given every lap. In this case, we're actually going to look longitudinally across days. So the mice, go through the standard 4 lap, one reward per 4 lap experiment. The second day, the reward is given every single lap. So it's kind of like a extinction paradigm. And we can see here that, as you might expect, these cells kind of lose their lap preference. They're not very well correlated anymore, but they maintain their spatial preference, of course. So this is a little bit more nuanced experiment. So here we're going to change the relationship between the events, where the mice first learn the standard 4 laps per reward schedule. And on the second day, a 5th lap is added, such that now the mouse has to run 5 laps before they can get their reward. So what will happen to the ESR preference of these neurons? Will it be able to recognize the addition of the 5th lap? And so what happens? So we can see actually that lap 1 and 2 cells stay the same. So they continue to prefer lap 1 and lap 2. Lap 3 is kind of lost. And lap 4 is still pretty well correlated, but there's a little bit more nuance to that. So again, this is just kind of a zoom in of lap 3. So we can see lap 3 is no longer really well correlated. So cells that preferred lap 3 during the first day no longer seem to on the second day. But what about lap 5? So first of all, looking at the lap 3 cells, a number of them preferred lap 3 on day 1, but then on day 2, they preferred lap 4. So there's this kind of sequential shift as the mice recognize they have to run 5 laps instead of 4. The preference of these cells actually shift sequentially. And it's interesting that, for instance, like it's not just totally reshuffled. So it's not that lap 1 cells are becoming lap 4 cells, it's 3 to 4. And then actually also the Lap4 cells are kind of in between. So we see here that the Lap4 cells are highly correlated as far as their increase in activity during Lap4. But actually, if we look also at Lap5, we see on day 4 that Lap4 activity is also elevated, but they actually are, many of them are shifting to prefer Lap5 instead. So showing, presumably that they're shifting kind of in relationship to the reward schedule. Kind of the salience of the proximity to the reward is potentially driving this shift. So again, looking at the proportion of cells that shift from Lap4 to a Lap5 preference, even though, as we said, it's not entirely exclusive, only a very small number are maintaining their Lap4 preference across days. So by changing the relationship between the number of laps and the reward, the ESR preference of these neurons is changing. So the next couple experiments are looking at this phenomenon, the fact that both the spatial preference and this event-specific preference exist within the same neurons. So to try to understand whether these are separable or related in some way, the authors injected halorhodopsin in the medial entorhinal cortex, which has been implicated in this kind of sequential event structuring in the brain, and then continued to monitor calcium activity in the hippocampus, and they were able to activate terminals, inhibit terminals from the medial entorhinal cortex during the course of the trials. So in this case, we have the standard experiment structure with 4 laps with a reward on the first lap. But this time, these mice alternated between 2 trials with the light off and 2 trials where input from medial entorhinal cortex was inhibited. So here's a couple of example cells, a Lap3 and a Lap4 cell. So we can see here that this Lap3 cell, preferred Lap3 in the absence of inhibition. But when input from the medial entorhinal cortex was inhibited, this ESR relationship was disrupted, and it kind of became more of a Lap2 cell during this case. But importantly, you see that whether the light was on or whether the light was off, the spatial specificity of the neuron stayed the same. So whether it's being activated during Lap3 or whether it's being activated during Lap2, it's still being activated in the same location of the maze. So similarly here, this is an example of a Lap4 cell. It's maintaining its spatial specificity, but its lap specificity is shifting in the presence of this optogenetic inhibition. This is just a summary data of the same thing. So again, looking whether between the light off or on, we can kind of see this same spatial pattern of neurons being maintained. But the ESR correlation is not as well preserved versus controls. So we might see, pay attention to the change in the y-axis here. So these are control mice that just had M-cherry injected into the medial entorhinal cortex. We can see that they are significantly correlated in the negative controls, but no longer in the optogenetically inhibited mice. But spatial relationship means highly conserved. So this phenomenon is both spatial rate remapping and the event-specific rate remapping can occur within the same cells, but they can actually be manipulated separately. We've seen this in a variety of different ways, whether it was with the square to circular maze, whether it was with this inhibition experiment, or in the changes in the spatial trajectory, kind of all are aligning and showing us the same thing, that these two phenomenons are kind of superimposed, but they are not really dependent on each other. But still kind of investigating what about other continuous variables. So what happens when time, rather than spatial location, is the continuous variable that's changing? So this is very similar kind of structure to the standard experiment that we see, but this part of the maze now is a treadmill, actually. So the mouse each lap is running on the treadmill for 12 seconds at a rate of 14 centimeters per second, I believe. And then it has to run around the maze again, and gets the reward at the start of the first lap each time. So here's just an example, our little mouse buddy. And so we can see here that just like hippocampal neurons represent this spatial rate remapping, so I should mention here, we're looking here at dynamic activity only at the period when the mouse is on the treadmill. So when the mouse is on the treadmill, we also see kind of time-specific rate remapping. So the mouse is staying in the same physical location, right? But as time proceeds, time is kind of represented by various cells. So this is just an example of non-event specific cells, but that are rate remapped in relation to time that the mouse is on the treadmill. Excuse me. So again, we're looking at a similar figure here to what we saw in the first figure, but instead of spatial location being represented on the x-axis here, we're looking at the time that the mouse was on the treadmill. And we see that this event-specific rate remapping phenomenon persists even when time becomes the salient continuous variable. There are cells that have this time dependent rate remapping kind of overlaid with this event-specific rate remapping. So in lap 1, 2, 3, 4, similar to what we've seen with the spatial location. And again, more individual examples here of like a lap 1 specific cell, 2, 3, 4, so on. Very familiar based on this spatial location. And this is just looking at the proportional breakdown, a little bit less common phenomenon than with the spatial location, but a similar phenomenon still. And side note that the treadmill has the added benefit of keeping the mouse's walking speed and head orientation constant. So they didn't actually have to use the model corrected delta F over F activity here. So they can just use the raw fluorescence, which is nice. So we can see that this stays constant even when time becomes the more relevant continuous variable that's changing. And just again, to kind of drive the point home, this is still tracking the relationship between events. So we're going to repeat our kind of negation experiment again with the reward being delivered each trial. And again, we see the same thing that we saw when we did this with the spatial, with this, just the standard laps around the maze in the absence of the treadmill. So now the event-specific rate remapping phenomenon diminishes demonstrably. And there aren't very many cells that exhibit this phenomenon anymore. So that concludes the papers. I'll just review some of the main conclusions. So about 30% of CA1 pyramidal neurons exhibited this lap preference, which we termed event-specific rate remapping. This phenomenon is transferable across experiences, which we saw as across days, cells maintained their ESR activity even when the maze shape changed, or the maze was elongated, or the spatial trajectory changed as the mouse had to turn left or right. These continuous variables like space and time are also represented as rate remapping in these same ESR cells. But these two phenomena can be manipulated individually. So they're just kind of superimposed within the same neurons. And finally, event-specific rate remapping tracks the relationship between events. So whenever the events are made indistinguishable by delivering the reward at the end of each lap, the ESR phenomenon essentially disappears. It's sensitive to alterations in the kind of abstract relationship between events. So we saw this when there were 5 laps instead of 4, for instance. The ESR activity shifted. And it's robust against changes in the continuous variables whenever the abstract relationship is kept constant. So again, we saw this when the square to circle maze, for instance, and some other examples. So that concludes my part of the presentation, but I would love to take questions from the audience if they're available.
Great. Thanks, Kevin. That was really clear and a really nice study, I think. Actually really clever set of experiments. I know we were talking earlier before we went live, just a really clever set of experiments to kind of probe the different aspects of this relationship between continuous variable representations and episodic event-based representations. I thought the treadmill experiment at the end was pretty cool. Actually, I'd love to see like a supplementary video of that.
Yeah.
Also, just as a side note, in your introduction, in reviewing their introduction with the experience of dining in a restaurant, it made me really yearn to be back out dining in restaurants. Right now, a lot of us can't do that. So I didn't realize how much I miss eating out until I read this paper. So their introduction.
Yeah, absolutely.
Yeah, we did get several questions. So I'll start with the first one that came in. And I think so the question is, how long does it take to train the mice for this task? And does lap preference of neuronal activity emerge over time? I think I noticed some answers to this, but if you don't recollect, I can chime in too, but go ahead if you.
Yeah, so let's see. So I believe I remember a detail being listed in the paper that each trial of 4 laps took maybe 90 seconds, I think, and they did maybe 20 of these, I think, in a day. So I don't know, what is that, 30 minutes, something like that. And they do show kind of the trial specific activity here. So, oops. I don't really see, oops, this is the wrong one. I don't really see it developing over time so much.
Yeah, they comment. Yeah, they comment on this a little bit though, and maybe it's I did catch this as you were presenting, and I saw the question come in. I kind of took a look, and it does look like the training, at least in some cases, was like 8 days of training. I don't know if they really required that, but they do say that the proportion of ESR cells, at some point they say that the proportion went up from about 17% to 29% over that 8-day period, and they did notice the running speed increased on the 4th lap compared to the 1st lap towards the end of that training, sort of suggesting that they've also kind of learned the period of this, 4 lap and then reward kind of event or whatever.
That's right. I remember that now.
So anyways, so there is some sort of learning that goes on, although they don't really probe that, any further. It's sort of an aside. Cool. And then, another question is, did the authors examine whether a cell's lap specificity is related to the location of the cell's place field, especially for cells with place fields in the start box or maze endpoint? Just one side note, I think they excluded, I think they pretty much just excluded cells that, had activity specific to the start box, or if they mean like the reward box.
Yeah, so if we look at this figure, so they did see cells that were specifically driven by the reward, and those were excluded from any relationship with this neuronal activity. I don't recall whether they looked at this relationship between the spatial location. I think Lap1 cells were more likely to be somewhat proximal to the box where the reward was given, although they did exclude the reward only driven cells. But that's one detail that I remember being in the paper, but I don't know what proportion, but I don't remember them detailing any other spatial relationship between the lap and the location.
Yeah, I mean, certainly here in this figure that you're showing here, it looks like there's pretty good spread, place field preference across all kind of, lap 1, 2, 3, 4 preferring cells. So, but yeah, I don't remember them going into that in too much more detail. Okay, yeah, another question. I think this one might be something that we just kind of conjecture on a little bit. Do we predict interconnectivity between the lap cells? For example, do lap 1 cells communicate with lap 2 cells? Are lap 2 cells getting their spatial temporal info from lap 1 cells? Any thoughts?
I am woefully ill-versed in the microcircuit of the hippocampus, honestly. I don't really know how this could possibly be established or really understand it. But I mean,
it's hard to say.
Whether it's a direct or indirect connection, it might be possible. I think it's a fascinating phenomenon, especially the fact that it shifts sequentially when they added a lap, right? So it's like lap 3 cells becoming lap 4 and lap 4 becoming lap 5. That's clearly not chance, at least intuitively. So yeah, something's going on there. I would be fascinated to follow up on exactly how the microcircuit of like the plasticity and how this is happening. But I really don't know.
Yeah, and also some interesting questions related to that in terms of like you could imagine some downstream brain regions that are sort of collecting this information, from these different populations of sort of event preferring, lap preferring cells. Another question was why the overrepresentation of the ESR cells tuned to lap 1 versus other laps? Is it, potentially associated with proximity to the recent reward? Like they've just been rewarded and so forth. So, because you might, it doesn't seem like necessarily, and I was actually thinking about this, it's kind of funny not to have another example of sort of shelter in place activities, but my family and I, we run laps outside in our cul-de-sac to get some exercise. And so it's about 12 laps 12 laps for a mile. And so, I got to make sure that I'm counting each lap and staying, remembering each one. Like if I miss lap 4, then I'm all off, right? And so to some degree, you might think that you really want to track each one and you want the same proportion of cells representing that. So yeah, kind of wondering why, certainly consistently, there's a overrepresentation of the lap 1 cells in this case.
Yeah, I mean, this is all conjecture, but I mean, I have to imagine it has to do with the reward, which kind of really serves as kind of the punctuation mark. It's kind of the reference. So whether you could imagine where if they had done an experiment, where it's lap 1, to 10 instead of 4. And if just think about it, if you yourself were running this maze, right? Like you would know, okay, so I just started, I just got my reward. And then kind of like you were saying, kind of laps 2 and 3 and, 5, 6, 7 are all kind of the same. They're all basically indistinguishable from each other. You're kind of keeping track of it. But then like when you're getting close to the finish line, right, you get a little bit more excited. And they even noted, as you mentioned earlier, that they might start running faster on the final lap, right?
Right.
So
Yeah.
I think, you do want to you don't want to lose count, right? But it's the beginning and the end that's kind of the most relevant and the most distinguishable from each other.
Yeah, that's right. Yeah, and sort of adding to that sort of question about the reward, it does seem like tracking the timing of this task, it needs to be behaviorally relevant. Otherwise, these ESR cells tend to sort of disappear for the most part. So, for instance, when they give a reward on every lap, you kind of see this whole thing abolished. So, one interpretation of that is that there's no relevance. That like the mouse doesn't need to track laps anymore, right? Because you're just getting a reward each time. But yeah, I was thinking it might have been interesting to do a different variation where you basically make the reward unpredictable, sort of randomly given, give it randomly at in some way, so that technically, you get rid of this timing cue based on the reward, but then you maybe use some other sensory cue, like an auditory tone, at the beginning of each of these 4 laps or whatever, or an auditory tone, and then every 4 laps the auditory tone again. So technically the sensory information would be there, but it's not particularly behaviorally relevant to the animal because they're being randomly rewarded or every time. And kind of just wondering, in that situation, would you still sort of passively see it encoded in the hippocampus because it's information, it's there and it just gets tracked, or because it's just not important truly to the animal, it would likewise just kind of reduce the number of ESR cells that you could observe?
Yeah, that would be very interesting. If you have these kind of 2 competing signals, I guess you'd have to figure out, exactly what is super interesting and what is uninteresting to a mouse. Because that isn't always so straightforward.
Right.
But
Yep.
yeah, I guess I would predict you might see kind of how would it do that? Maybe the mouse, because like you certainly can track 2 different things at once. So you could imagine if like every 4th, time there's a reward, and then every 6th time there's something that totally not relevant, but you would still be able to count each of those 2 things separately. So then like, would you see kind of 2 populations arise? Like, yeah, that would be kind of really interesting.
Right. Yeah, there's quite a bit like it seems like could be interesting to do with sort of varying the reward and like the value of the reward and things like that. But anyhow, yeah, just one last question, I guess, and I don't actually know offhand. I don't know if you do, since neither of us are necessarily place cell experts and hippocampus experts. But someone was wondering how does 30% lap modulated cells compared to, or ESR cells, if you want to call them that, compared to just like place preference cell proportion?
Oh, I do know that. They mentioned in the paper that they saw 72%, I believe, was the number of cells that that were just like this. They were, spatially rate remapped, but not Oh no, sorry. I think the 70% includes both the ESR cells and any non-ESR cells, but that had this spatial remapping.
I see. Yeah.
So
Yeah, but you're right. I remember, yeah, okay. I do remember a higher, yeah, something around 70%, I thought actually for just general place cell activity in their population, but I could be wrong also. But I do think place cell proportions are higher certainly than the 30% that they see
yeah,
here. But
I think as I understood it, the 70% was any spatially rate remapped cell, including ESR cells, was my understanding. But I'd have to refer back to the paper to make sure I have that exactly right.
Yeah, yeah. Rather than, in other words, rather than a cell that's just always encoding that place regardless of lap number, which would be a potentially separate population of
Right.
cells. Or
Yeah.
Got it. Yep. Okay, great. Well, thanks again, Kevin. And if there are no more questions, we'll wrap things up. Thanks to Kevin for walking us through this interesting publication, and thanks to everyone else for attending and sending in questions. We hope this was a welcome scientific discussion for everyone, and that you'll join us again next week when we'll be going over another recent circuit neuroscience publication. And so until then, please stay safe and healthy, and hopefully we can all get back to our regular research activities, our regular restaurant dining activities very soon. So thanks, Kevin, and take care, everyone.
All right. Thanks, everyone.